My name is Mike Leone. I'm a math major from New College of Florida, and I worked with Dr. Fink this summer at Ohio Wesleyan University, and we studied neuronal synchrony of scale-free networks. And the reason we're interested in synchrony of neuronal networks is that it's been associated with directed attention, memory formation, and dysfunctions such as Parkinson's disease, autism, and epilepsy. We were interested in the degree of synchrony of a system. And we focused really on two different features. One was the network connectivity, so the structure, the way that neurons are all connected. And we also looked at individual characteristics of a cell. In terms of individual characteristics, you can classify a neuron as type 1 or type 2 based on the way it responds to a pulsive current. A type 1 neuron, it'll always fire sooner once it receives a pulsive current, so it always speeds up. A type 2 neuron will sometimes speed up and sometimes delay in firing. So with these two different characteristics and classification types, we were interested in networks that had type 1, type 2, and we were most interested in when they were mixed. In terms of network structure, we were focusing on what are called scale-free networks. We're motivated by recent evidence that highly connected cells called hub cells develop in the mouse hippocampus. These hub cells persist into adulthood and have been shown to orchestrate synchrony. They're highly influential in the system. In order to model scale-free networks, we created them using the Barabasi-Albert algorithm. Basically, you start with a small, fully connected network, and then as you add neurons and edges, the probability of a pre-existing neuron receiving a new edge is proportional to the number of edges it already has. You could think of it as a rich gets richer scheme. As a comparison, we also studied what are called small world networks. These networks have a spectrum starting with local connectivity. So in other words, you connect the neurons within some radius to nearby neurons. With some probability p, you take each edge and you reassign it randomly to some other neuron in the network. So for example, with p equals 0, you stay with a local network. But with p equals 1, since you're reassigning every edge, you're going to get a completely random network. And so the small world network is a description of that spectrum in between. We modeled our neurons using the Morris-Lacquard model. It's a set of differential equations that ultimately describes the change in membrane potential over time of a neuron. And so how you get that spiking behavior in response to current. The Morris-Lacquard model is helpful because it gives us both type 1 and type 2 neurons, and all we have to do is change a single parameter in order to get either type 1 or type 2. And furthermore, we can control for synchrony using the Morris-Lacquard model because as you change the current, the rate of change of frequency is the same for both type 1 and type 2 neurons. So for our first simulations, we did isolated networks, either type 2 or type 1, and scale-free. We found that for the type 2 networks, we got high synchrony. And as we increase connection strength from neuron to neuron, the type 1 networks, they lost synchrony. So we saw in this region a huge difference between synchrony, desynchrony between type 2 and type 1. This is actually a preliminary result. We really expected to see the type 2 neurons synchronize. The type 1 networks, we didn't expect them to synchronize at all. So going forward with that information, we started to mix networks. So what we did was we had a whole spectrum of percentage of type 2 neurons in a network, going from fully type 1 to fully type 2. And for every simulation, we measured synchrony. We saw that as we added type 2, in general, the synchrony increased. If we added the type 2s randomly, that is, assigned type 1s and switched them to type 2s randomly, this black curve here, it increased relatively linearly. If we purposely assign the type 2s to be the hub cells, so we pick the neurons with highest degree to be type 2, then immediately we would see increases in synchrony with a small amount of type 2. So there was a drastic increase. If we purposely picked the type 2 neurons as the least connected cells, then we actually saw synchrony would stay relatively low until you've reached some threshold of over 60% type 2 neurons, and then it finally synchronized. So seeing these different behaviors between the way we assign the type 2s, you could think of the red curve as being assigning the hub cells to be type 2, and what were to happen if the hub cells were type 2. Going back to my mentioning of hub cells being in the hippocampus, we're not actually sure what type these hub cells are in terms of excitability. As a comparison, we ran similar simulations with small world networks. We wanted to see the way in which synchrony increased as we added type 2s. And so in general, there was yet again an increase for small world networks. But the way it happened was the synchrony stayed relatively low until the majority of the network was type 2. And then we finally saw increases in synchrony. For the scale-free results, we saw increases right away, which suggests that in scale-free networks, individual neurons have more influence over the system. And finally, we ran simulations where our hub cells, we actually switched them to be inhibitory neurons. So rather than exciting neurons around them, they would tend to silence them. And this is in agreement with experimental evidence that the hub cells of the hippocampus tend to be inhibitory. So by setting the 20% most highly connected cells, or the hub cells, as inhibitory, the rest of the network as excitatory, and all excitatory cells as type 1, we were interested in what if the hub cells were type 1 versus type 2. So running these different simulations, we came up with very different results. When the hub cells were type 1, the system couldn't synchronize. It stayed relatively random in the way it fired. But if the hub cells were type 2, we saw synchrony and a great increase in the firing pattern in the way that they all fired together. So from that, there's some evidence to suggest that the hub cells in the hippocampus that are known to cause synchrony, there's some evidence that they're type 2. And so that was my work this summer. And I'd like to thank Dr. Fink for his guidance. And I'd like to thank the National Science Foundation and the REU program and Ohio Wesleyan University.